SIV-HIV VPU functionality is microevolution. It does not demonstrate mega-evolution. One of our viewers requested, "I want a video about evolution of SIV to HIV, in which VPU of SIV cannot antagonize Tetherin, and that SIV, VPU evolved to VPU in HIV and can antagonize Tetherin." Our response is as follows. Thesis: SIV-HIV VPU functionality is microevolution. It does not demonstrate mega-evolution. Details. 1. Microevolution is adaptation of a species inside a genus or family. This can occur through random chance and natural selection. Mega-evolution is the formation of completely new body plans, and completely new higher taxa, kingdom, phylum, class, order. Random chance and natural selection cannot create mega-evolution, as we have discussed in previous videos. 2. SIV-HIV VPU change is a change inside a virus, and a closely related virus, so it is microevolution at best, which is not in question. 3. This is not an evolution of a new function because some forms of SIV already have a VPU protein that antagonizes Tetherin. So, this is not a new function to SIV. Scientists believe that HIV came from SIV. A SIV that has Tetherin antagonism is more likely to be able to zoonotic switch to humans and cause HIV, and then spread among humans. 4. So, at best, this is a loss of a function, by mutations, followed by a regaining of that function, by mutations. Viruses have the highest populations, in the biosphere, and the highest mutation rates, apart from viroids, and so they can regain a lost function as long as there are no more than 20 specific functional mutations needed, as an upper bound. 5. The estimate for the SIV-HIV VPU is 3 amino acid position changes. So it is within the capability limit mentioned above. 6. In addition, viruses can perform horizontal gene transfer, from one virus to another, or from virus to host, and from host to virus. So, the Tetherin antagonistic functionality can be transferred from one virus that has it, to another virus population that does not have it. 7. Even if a new function was generated, which it was not, that would not invalidate the concept of irreducible complexity, IC, because IC requires us to consider the probabilistic resources of the biosphere, as a lower bound, or the universe, as an upper bound, to determine if a system is irreducibly complex. See below. 8. That is, if the parts complexity of a system, or machine, is below the probabilistic resource limit of the universe, we do not define the system as being irreducibly complex. However, if the parts complexity of a system, or machine, is above the probabilistic resource limit of the universe, then and only then, do we define that system as being irreducibly complex. 9. We have applied this protocol, above, to orphan genes in several of our previous videos. The universe can discover, by random chance and natural selection, no more than 3 completely new unique high-specificity proteins and genes, or 12 completely new unique lower specificity proteins and genes, in its entire 14 billion year history. 10. We have calculated this probabilistic resource limit, or material and time limit, for the universe, 10 to the power of 120 chemical trials. 11. Irreducible complexity just means that a system has a certain minimum level of complexity, parts that fit together in a certain way to enable the target function of the system. So, the number of non-functional, random, mutations needed to create a particular functionality will determine if a particular nanomolecular machine is irreducibly complex or not. See below. 12. To determine the edge of evolution, i.e., the limits of what evolution can accomplish, we have to look at the population size and mutation rate of the population. That is why, in our videos, we always refer to the material resources of the physical universe in its 14 billion year history, as a maximum of 10 to the power of 120 chemical trials. And that is what would determine what is possible for the universe to accomplish with random chance and natural selection in its entire 14 billion year history. That limit is 3 high-specificity orphan proteins, or 12 low-specificity proteins. 13. This is why microevolution is possible. Adaptation of a species inside a genus or family, including low-level speciation inside a genus or family. I.e., the number of genetic and morphological changes needed for microevolution are within the reach of evolution, random chance and natural selection, given the population size, mutation rate, and time available. 14. And, this is why mega-evolution is not possible. Creation of enough orphan genes and gene regulatory network instructional segments for the creation of completely new body plans, and completely new higher taxa, kingdom, phylum, class, order. I.e., the number of genetic and morphological changes needed for mega-evolution are not within the reach of evolution, random chance and natural selection, given the population sizes, mutation rates, and time available, e.g., the 14 billion year history of the universe. 15. Note: this math and reasoning shows that humans did not evolve from an alleged chimp-human ancestor by random chance and natural selection. There is simply not enough time even in the entire 14 billion year history of the universe for all of the human orphan genes and proteins needed to arise by random chance and natural selection. This is the case for all of the higher taxa: kingdom, phylum, class, order. Conclusion: SIV-HIV VPU function is microevolution. It does not demonstrate mega-evolution. Thanks to Mena Sedrak for the question. Thanks for watching. Please like, share, and subscribe.